Welcome back. This lesson is on Dihybrid Cross, and the learning goals to be achieved from this session are: 1. Illustrate Mendelian Dihybrid Cross up to F2 generation. 2. Complete Punnett Squares for dihybrid crosses. 3. Predict phenotypic and genotypic ratios for dihybrid crosses based on the Punnett square. And 4. State the Mendelian law for inheritance of two traits. In the previous lesson, we learned about monohybrid crosses, where Mendel studied the inheritance of one trait in pea plants. From his several thousands of the monohybrid crosses, Mendel formulated two laws of inheritance: law of dominance and law of segregation. He proposed that the pair of factors for each of the 7 contrasting traits in pea plants exists as a dominant and a recessive factor. And these pair of factors in the parent segregate during gamete formation and results in the formation of a consistent 3:1 phenotypic ratio in the F2 generation. Now, Mendel's work does not end here. He expanded his hybridization experiments in order to study the inheritance of two traits. And he did this by crossing two true breeding pea parental plants that differed in two traits. However, the basic steps of hybridization remain same to that of monohybrid cross. Since you have already worked out on the monohybrid crosses, I'm pretty sure that you will find it easier to follow through the steps of dihybrid cross. Let me now give you an overview of Mendelian dihybrid cross by taking an example of a cross between round and yellow seeded plant with a wrinkled and green seeded plant. So in this cross, the two parents differed in seed shape and seed color. Now, we must also remember that Mendel always selected 2 breeding lines for his parental generation. So in this cross, one of the parental line had round and yellow seeds, and the other parental line had wrinkled and green seeds. The cross between these 2 lines resulted in F1 dihybrid plants that had all round and yellow seeded. So F1 plants were all showing round and yellow seeds. Now, this result showed that the dominance of round over wrinkle and the dominance of yellow over green is not affected by the inheritance of the two traits simultaneously. And after attaining his F1 plants, Mendel then allowed the F1 plants to undergo self-pollination. And he then was able to subsequently obtain his F2 generation. And when he looked into the phenotypes of the F2 generation, he observed 4 different phenotypes in the F2 generation in a ratio which was 9:3:3:1. So out of the 16 possible combinations, what he saw was 9 of them had round and yellow seeds, 3 of them had round and green seeds, yet another 3 had wrinkled and yellow seeds, and 1 had wrinkled and green seeds. So this was his typical F2 phenotypic ratio, which was 9:3:3:1. To confirm his result, Mendel conducted dihybrid crosses for other combinations of traits. And from several thousands of the other dihybrid crosses that he conducted, he found a consistent F2 phenotypic ratio of 9:3:3:1, similar to that obtained for seed shape and seed color. So what could be the possible explanation to this pattern of inheritance? To understand how Mendel explained dihybrid results, we first need to work out the cross up to F2 generation. And for this, as we have learned, the first step is to assign letters to the traits. And here we have 2 traits to be considered simultaneously: seed shape and seed color. In the seed shape, through Mendel's experiments, we know that round is dominant over wrinkled, and in the seed color, yellow is dominant over green. Now, we also know how to assign letters to the respective dominant and recessive factors. So uppercase letters are to be used for dominant factors and lowercase letters are to be used for recessive factors. So let's first write down the letters for the trait. let round be capital R, let wrinkled be small r, right? Likewise, let yellow be capital Y and let green be small y. So I finished assigning the letters for the respective traits. Now we want to be starting with P generation. So P generation, as we know, are true breeding parents. So we have, the parents are, one of the parents have round and yellow seeds. So one of the parents have round and yellow seeds. The other parent have wrinkled and green seeds. So let's consider this as a male parent and this as the female parent. Now, because we know parents are always true breeding lines, so it becomes easier for us to be assigning with the genotype for these two parents. So since we have given capital R as the allele or the factor for round and capital Y as the allele or factor for yellow, so now the genotype for this parent has to be, and we know that each of the traits should be having two alleles. Alright, so this trait should be having two alleles, but it has to be identical. This trait has to be having two alleles, but it has to be identical because the parents are true breeding round and yellow seeded plants. So I'm going to be assigning the genotype here two capital R's to denote to the round genotype and two capital Y's for the yellow genotype. Right? So in this way, each of the parents are going to be having, in a dihybrid cross, they are going to be having 4 factors. Okay, unlike the monohybrid cross where we had 2 factors in each of the parents, here we have 4 factors for each of the parent because we are considering 2 traits at a time. Right? Now, for your convenience, I'm going to be using another color for the other parents so that you can be having a convenience in tracing with the parental gametes. So this parent has wrinkled and green seeds. So we know wrinkled is small r, green is small y, but then because we have to have 2 alleles or 2 factors for each trait, so wrinkled would be, and because the parent is homozygous, it's going to be small r, and green would be small y, small y. So in this parent, we have 2 of the homozygous alleles for the traits, and the genotype becomes small r, small y, small y. So again, it has 4 factors. The next step is to show gamete formation, right? So how we have the gametes released from both of these true breeding parents. Now each of the parents have 4 factors. Right? 2 factors for each of the trait. So now, from our understanding of the law of segregation, gametes are to be receiving one of the 2 factors, right? But because we have 2 traits here, we must remember to make sure that each of the gamete receives one of the factors of each trait. Okay, so each of the gametes should be having one of the factors of each trait. So in order to make it more simpler for you, I'm going to be having the 2 alleles or the 2 factors of round as R1 and R2. Likewise, the 2 factors for yellow as Y1 and Y2. Okay, the first Y and the second Y. Now, how we can be having the segregation of the gametes or how we can be having the possible combinations of these factors in the gamete is we can have the combination of a gamete with the first R with the first Y, which would be a gamete that is going to be RY. Okay, likewise, we can also be having the first R combining with the second Y and forming another gamete, which is also RY. Likewise, we can be having our second R combining with first Y and resulting in another RY. And then we can be having the second R going with the second Y and forming another gamete with RY. But what you see here is, even though we have 4 numbers of the gametes or 4 possible gametes, but each of these gametes are basically having the same kind of genotype, alright, because the parents are true breeding. So whenever we have true breeding or homozygous parents, we are to be achieving or attaining identical gametes. So we have 4 possible gametes from the females, but then they are all identical. Likewise, we can look into the gametes produced from the male parents, which have wrinkled and green seeds. Now, such a parent has 2 small r's and 2 small y. So we can have the first r going with the first y, forming into a gamete, right? So each of the gamete receives one of the factors of each trait, and then we can have the second R going again with, or we can say, sorry, the first R going with the second Y. Then we can take the second R going with the first Y, as well as the second R going with the second Y. So all of these are possible combinations of gametes. So what you see here again is we have 4 possible gametes, but then because wrinkled green is a homozygous genotype, we are always to be achieving 4 identical male gametes, right? So this is the step which is gamete formation. Now then we have to show F1 generation, first filial generation, and first filial generation again has to be shown in terms of all the possible combinations of genotype, and for that Mendel used Punnett square, right? So we know Punnett square is the grid that we want to be representing here, and the number of Pritts should be, as I said earlier, should be determined by the number of gametes. So we need to be having this time in the monohybrid crosses we had 2 by 2 kind of the Punnett square, but here since we have 4 gametes from each of the parents, so we are to be drawing 4 by 4 Punnett squares. Okay, so let me draw the Punnett square. So I need to be drawing 4 columns this time. Okay. And then I should be having 4 rows as well. Okay, so once I'm done with this Punnett square box, now I have to place the parental gametes. So we know we are going to be placing it one on top, one of the parental gametes on the top and the other parental gametes on the side. Okay, so let me place all the male gametes on the top. So I have all of the male gametes which are small r and small y, right? Then I'm going to be placing all of these female gametes here on the left side, and all of the female gametes are also the same type, capital R, capital Y. Using Punnett squares, we are able to predict all of the possible genotypic combinations of a cross. Now for this dihybrid cross, we have a possible 16 different genotypic combinations for the cross. So I'm going to be now completing each of the genotypic combinations by combining the respective parental gametes one by one. But before doing that, what I want you to keep in mind is when we are writing down the genotype, we are to be writing the same letters of a trait together. What I mean by this is, for example, here we have to combine capital R, capital Y with small r, small y in this box. Now while making the combination, we will never be writing the combination like this. Capital R, capital Y, small r, small y. This is not the way to write a genotype. Okay, because we have to be always writing the letters of the same trait together followed by the next second trait. Okay, so how we are going to be writing is, we are going to be writing capital R, small r, followed by capital Y, small y. So this would be the correct way to write the combination of capital R, capital Y, female gamete, and small r, small y, male gamete. Likewise, now we can be completing the rest of the other boxes. So the second box, or in the second column, we have to have the female capital R, capital Y, combining with male small r, small y. So we are having, and always we need to be writing the capital letter first. Okay, so capital R, small r, capital Y, small y. Okay, in the third box, it would be capital R, small r, capital Y, and small y. Okay, then in the fourth one, we are going to be having capital R, small r, capital Y, small y. Okay, so we completed with the first row here. Now in the second row, again we are having female gamete is the same type, which is capital R, capital Y. Male gamete is small r, small y, is the same. So what we can be guessing is, basically we are to be achieving the same genotype, because you have the same gametes always contributed from the male as well as female parent. So this is how we complete with all of the 16 boxes with the possible genotypes, right? Now we are going to be making the observation of the genotypes. So what about the F1 genotype? So what is F1 genotype? Now here, although we have 16 possible combinations, but all of the combinations are the same combination. So we have all capital R, small r, capital Y, small y. 100% of the F1 plants are having such kind of a genotype. And this genotype, as we know, is not homozygous, because the factors for round color is capital R, small r, and the factors for Y color is capital Y, small y. So it is heterozygous. And such kind of plants are called as dihybrid plants. Why dihybrid? Because they are heterozygous for two traits. Okay, so the plant here is heterozygous for round as well as heterozygous for yellow. And because it is heterozygous for two traits, it's called as dihybrid plant. Okay, so we obtain a dihybrid plant whose genotype is always capital R, small r, capital Y, small y. Okay, so that was how Mendel came down to the F1 generation and its genotype. And the phenotype, what about the phenotype of such F1 plants? Phenotype, Mendel could be easily observing all of the F1 plants were yellow, round and yellow. Round and yellow seeds. So 100% of his plants had only one phenotype, which was round and yellow seeds. Alright, so this is how we have come down to the F1 generation. Now after this, the next step is to do F1 selfing. Now, as we know, F1 selfing is basically to have a kind of self-pollination within the same F1 plants. But then we can be showing the two sex cells where both of these cells are going to be heterozygous for round and yellow, right? Because we are using the F1 plants. So now, let's have the gametes of the F1. So the next step is to show the gametes from such F1 plants. Now, what would be the possible gametes from such F1 plants? Now, here the gametes are going to be a little more complex. Okay, so we need to pay a little more attention here. Now, as I said, we have to have segregation of the pair of factors during the gamete formation, and each of the gamete is to be receiving only one of the two factors for each trait. Okay, so now here we have two of the different R's and two of the different Y's. Okay, so the R, capital R, first R is capital R, second R is small r, first Y is capital Y, second Y is small y. Okay, so in that kind of a combination, how we have the possible gametes from each of these female and male sex cells. So we can be having 4 possible combinations of gametes from each of the male and female sex cells. So what about the female gametes? The first female gamete can be first R with the first Y. Okay, so each gamete should be receiving only one of the factors of each trait. Okay, so first R with the first Y, resulting in an egg having capital R, capital Y. Then let's have the second egg, and the second egg would be first R can also be combining equally. It has a 50% chance of combining with small y. This is the second type of egg. The third possibility is second R can also combine with the first Y, having a small r,
capital Y
type of egg. Okay, and the last possible combination is second R and second Y, which is small r and small y combination. So in this way, from the female cell, we are able to achieve 4 different types of gametes. Now please note, in this from the F1 plants, we are able to be achieving 4 different types of gametes. The reason is because parents are heterozygous. So when the parents are heterozygous, we have dissimilar gametes produced from the parents. When the parents are homozygous, we have identical or similar gametes all the time produced. Right? So now here we have 4 dissimilar eggs produced by the female cell. Likewise, we can be having 4 different types of male gametes even produced from this male cell, because it's even heterozygous. So it's going to be again capital R, small capital Y, first R, first Y, resulting in RY, male gamete. Likewise, first capital R with second Y, which is capital R, small y, which is the second male gamete. Third male gamete would be small r and the first Y, capital Y, which is small r, capital Y, male gamete. And the last one is second R with the second Y, which are both small r and small y. Okay, so in the same way, you have 4 of the gametes obtained from F1 male sex cell. So in this way, we have 4 different types of gametes produced from each of the, you can say, the sexes. Okay, and as a result of which, here we have to again have, when we are showing F2 generation, we need to be again having 4 by 4 Punnett square. In order to predict with all the possible genotypic combinations for F2, we need to be drawing Punnett squares which have 4 by 4 squares. right? So let's draw the Punnett square where I need to be having 4 columns and 4 rows, right? And after that we are going to be placing the gametes, right? So the gametes are, oh sorry, I forgot to use the color. So the gametes are, let's say male gametes. I'm going to be drawing it on the top of the grid. Capital R, capital Y. Second one is capital R, small y. Third one is small r, capital Y. Fourth one is small r, small y. So these are the 4 different types of male gametes. Likewise, we have 4 different types of female gametes which are to be shown here on the left side. So we have the first one is capital R, capital Y. Second one is capital R, small y. Third one is small r, capital Y. Fourth one is small r, small y. Alright, so we have the female gametes shown here on the side, and we have the male gametes which are shown here across the grid. Okay, now what we need to be doing is we have to be completing the Punnett square. In another way, this is basically nothing but we have to make all the combinations of the respective parental gametes, right? This is in a way just trying to show all of the random chances of fertilization, right? So we can be having any kind of the random fertilization taking place, and all of these, each of these gamete has an equal chance of combining with, each of the female gamete has an equal chance of combining with any of the other male gamete. Okay, so now let's take each of the boxes one by one. So the first one here is capital R, capital Y, and the first on the male side is also capital R, capital Y. So the combination would be becoming capital R, capital R, capital Y, capital Y. Both homozygous. Then the second combination would be capital R, capital R. Okay, so let's try to be doing it more systematically so that we don't get to make any kind of a mistake while noting down the combinations. So capital R, capital R. Next we are going to be having Y. Capital Y and small y. Okay. Third one, capital R, small r. Right? So capital R and small r. Then capital Y, capital Y. So we have capital Y, capital Y. The fourth combination is capital R, small r. Capital R, small r. Capital Y, small y. Capital Y, small y. So it just calls for a little of your attention and patience. Next one, capital R, capital R. Capital R, capital R. The second one here is small y. There it is capital Y. Now do we write small y and capital Y, or should we be writing capital Y and small y? I hope you remember. Yes, you're right. So it has to be not small y, capital Y, but it has to be always, the dominant factor has to be always written first. So it has to be capital Y followed by small y. Okay, so you need to keep this in mind. This is also a kind of a general rule where we say that when we have two of the letters to be written for a trait, always the dominant factor has to be, or the letter has to be written first, and then you can be having the recessive factor. Then next one is capital R, capital R, capital R, capital R, small y, small y. Small y, small y. Next one, capital R, small r, capital R, small r, small y, capital Y again. Okay, so I need to reverse this with capital Y first and small y. Right? And the last one is capital R, small r, capital R, small r, small y, small y, small y, small y. So here we are finished with the second female gamete having equal chances of fertilizing or combining with any of the male gametes, 4 different male gametes. Likewise, we'll go with the third female gamete. Okay, now this third female gamete has equal chances of fertilizing with any of the male gametes, which we are going to be showing in this row. So now small r, capital R. So we have to first write capital R. So capital R, small r, and capital Y, both capital Y. Second one, small r, capital R. Right? So it has to be capital R, small r. I hope you are noting this. Right? So we have to be writing the first capital letter first and then followed by the small letter. Right? You will not be writing small r and capital R. That's not the normal way to write it. So capital R, small r, and capital Y, small y. Capital Y, small y. Okay. Next one is small r, small r. So it is small r, small r. Small r, small r, and capital Y, capital Y. Capital Y, capital Y. Okay. Then the next one is small r, small r. Small r, small r, capital Y, small y. Okay, I'm using the colors just to give you a kind of a better understanding with my subsequent discussion. So don't bother about this. You can be writing with the same color. Then next we are to be taking the last female gamete, making equal kinds of possible combinations with the 4 male gametes. So let's complete the genotype. We have capital R, small r, capital R. But we need to be writing capital R, small r. And then it is capital Y, small y. Even though it is small y, capital Y, I'm going to be writing capital Y, small y. Okay, then next one, small r, capital R. Small r, capital R, or it has to be capital R, small r. Then it is small y, small y. Right? Small y, small y. Small y, small y. Okay, then we have next one is small r, small r. Small r, small r, small y, capital Y, which is to be written as capital Y, small y. And finally we have small r, and small y, small y. Small y, small y. So this is how we can be noting down with all of the possible genotypes by using Punnett square. Right? So again, what you find here is from the Punnett square, we learn that there are 16 possible genotypes that can be achieved from this cross. And from the Punnett square, we are also able to be predicting with the frequency or the ratio of these genotypes as well as the ratio of the phenotypes. With this, we have completed with all of the 16 possible combinations of genotypes for F2 generation. As we know, Punnett square is used to predict with the outcomes of genetic crosses, or we can say this is used to predict with all the possible genotypes of a cross. Now, Punnett square applies the rule of probability. So here what you can be seeing is all of these gametes, each of these gametes. For example, let's say the female gamete having capital R, capital Y has an equal chance of fertilizing with any of the 4 male gametes, right? And because of which we can be expecting all such 16 different combinations of genotypes through random fertilization. right? So we can be just having some kind of a basic idea like each of this has an equal chance of fusing with any of the other gametes of the opposite parent. Likewise, each of this male gamete has an equal chance of fusing with any of the other female gametes. Okay, so in that way we are able to be applying with the rules of probability. Specifically, we are looking into the products of the probabilities here. Now, let's try to study with the genotypes and the possible phenotypes of the F2. So what Mendel observed was, out of these 16 combinations, he could be finding 4 different phenotypes in his F2 generation. And the 4 different phenotypes can be easily predicted from the genotypes. Okay, so let's take each of the boxes one by one and let's try to predict with the possible genotype, because we know the kind of the dominance and the recessiveness of these traits or the factors. So now the first box has a genotype which is homozygous RRYY. So we can say that this would be having round yellow, right? So this is round yellow. The second one has capital R, capital Y, small y. And because it has one capital Y, again it's going to be yellow, right? So it is round yellow. Third one, capital R, small r. It has one capital R, so it is more than enough to be expressing its trait. And it has both of the dominant yellow alleles. So it's going to be yellow, round yellow. Capital R, small r, capital Y, small I. Now Y, so since they are heterozygous and have one of the dominant factors for each of the alleles, for each of the traits, so they are going to be becoming even round and yellow, right? So let's have all of the round and yellow. I'm going to be underlining all of the round yellow. This is also round yellow, round yellow, round yellow, round yellow, right? Then next one, capital R, capital Y, small y. It's going to be round yellow. Now this capital R, small y, small I is not round yellow, right? So let's skip that. Again we come to capital R, capital Y, small y. This is round yellow. Then this cannot be yellow. Next one, here we have another round yellow, capital R, capital Y. And the next one, capital R, capital Y, small y, heterozygous round yellow. And then the last one is here, capital R, capital Y, small y. So now if you count the underlined boxes, you can be saying you have 1, 2, 3, 4, 5, 6, 7, 8 and 9. All of these 9 of the boxes are having the genotypes which can be giving rise to. So if we have to look into F2 phenotypes, Mendel could be observing 4 phenotypes: round and yellow. So out of the 16 boxes, we have 9 having, out of the 16 boxes, 9 boxes have the phenotype which is round and yellow. Okay, the next one is, let's go into this one. Capital R would be round. Small y would be green. Okay, so it is round and green. So the next trait or phenotype is round and green. Let's look how many round and green we have in the box. So round and green, I'm going to be underlining with the green line. So round and green, then we have round and green. Okay, so green has to be always homozygous because green is recessive and it can be expressing only when they are present in the homozygous recessive state. So do we have any YY again? Yes, here we have small YY in homozygous state. So these are the 3 genotypes which are going to be resulting in round and green. So out of the 16 boxes, we have 3 that has round and green seeds. Okay, next we have this one, small rr. Small rr is, we know, small r is representing wrinkle, right? So small r means because it is homozygous for small r, it can be expressing wrinkled. So it is wrinkled seed, and capital YY is yellow. So we have the next phenotype, which is wrinkled and yellow seed color. And let's see how many of the wrinkled and yellow seed color we have. So I'm going to be just having a circle on the wrinkled yellow genotype. So we have the first wrinkled yellow genotype there with small r, capital YY. Okay, next one, small r, capital Y, small y, right? So we have yellow trait again. This is going to be wrinkled yellow. Then the other one that we are left with is these two boxes. So this one is small r, capital Y, small y, which is going to be wrinkled yellow. But the last one isn't. You don't have capital Y there. Okay, so now of the 16 boxes, we have 3 more that has wrinkled and yellow seeds. And the last box, this one, is having wrinkled and green. Okay, so it has wrinkled and green because it is homozygous recessive for both the traits. It is homozygous recessive for R, which is wrinkled, and it is homozygous recessive for Y, which is green. Okay, so the last phenotype that we are having here is wrinkled and green, which is one of the 16 possible combinations. So in this way, Mendel was able to be observing a very typical, consistent dihybrid F2 phenotypic ratio, right? Why I say it is consistent, because he carried out thousands of other similar dihybrid crosses for even other combinations of traits, and all of the results had this similar kind of a consistent phenotypic F2 phenotypic ratio, which is 9:3:3:1. Okay, and we can be easily finding with such kind of the 16 different possible genotypes and all of these 4 possible phenotypes by working on the Punnett square. Alright, so the phenotypic ratio is 9:3:3:1. This is very important for you to be noting. So Mendel had a monohybrid F2 phenotypic ratio, which was 3:1. Right, now we have Mendel's dihybrid F2 phenotypic ratio, which is 9:3:3:1. Okay, now for this cross, 9 is round and yellow, 3 is round and green, another 3 is wrinkled and yellow, and 1 is wrinkled and green seeds out of the 16 possible genotype combinations. Well, so we know Mendel's dihybrid F2 phenotypic ratio is 9:3:3:1. But what about F2 genotypic ratio and the genotypes? Well, for that, what we want to be doing is we want to go back to the Punnett square. Okay, and you can be easily predicting with all the different types of, or all the possible genotypes of the F2 plants from the Punnett square, as well as you can be also, if you work out on each of the genotype, you can be also knowing with the genotypic ratio in F2 generation. Now what I can be saying for you is there are a total of 9 possible genotypes. Okay, there are a total of 9 possible genotypes in F2 generation if you study the Punnett square. So I've given the number of genotypes, but I'm not going to be working out the type of genotype and the genotypic ratio. Okay, so that work would be your work. So what you're going to be doing is you're going to be noting down all of those 9 genotypes that I stated would be there for F2 plants, and then you can be even counting with or calculating with the genotypic ratio for the F2 plants. Okay, by looking or by studying the Punnett square. Now comes the big question. How did Mendel explain with this pattern of dihybrid inheritance? As we know, Mendel was very methodical and thorough with his experimental work. So what Mendel did was, Mendel analyzed and recorded all the numbers of the seeds with specific traits. He recorded or counted with the number of seeds with round shape, wrinkle shaped, yellow color and green color. And from all of these mathematical work and the counting of the seed numbers, what Mendel finally realized was that the 9:3:3:1 ratio was actually two independent 3:1 ratio that combined at random in order to form 9:3:3:1 ratio. So he was able to observe from his counting of the seeds that the number of round seeds to that of the number of the wrinkle seeds were of the ratio 3:1. So 3/4 of the seeds were having round shape and 1/4 of the seeds were showing with the wrinkle shape. Likewise, 3/4 of the seeds were showing with the yellow color and 1/4 of the seeds were showing with green color. So he could be realizing with two of independent 3:1 ratio, typical for each of the two traits of seeds in terms of seed color and seed shape. And these two 3:1 ratio independently had in a way assorted together randomly to form 9:3:3:1 ratio. Let me now give you a brief idea about how Mendel explained with the random combinations of two independent 3:1 ratios, right? Here we have the 3:1 ratio of round and wrinkle seed, and then we have on this side the independent 3:1 ratio of yellow and green seed. Now he said because of the random combinations of these two independent 3:1 ratio, it resulted in the formation of 9:3:3:1 F2 phenotypic ratio in dihybrid crosses. So how he explained was, if 3/4 of the F2 round seeds were to combine with 3/4 of the yellow seeds. Okay, so if 3/4 of the round seeds were to combine with 3/4 of the yellow seeds, this would be resulting in round yellow combinations. Okay, so if we look into the kind of the product rule here of a probability, so it is going to be resulting in 9 possible phenotypes that are going to be of round and yellow. Likewise, he said if 3/4 of the round seeds were to combine with 1/4 of the green seeds, because green seeds are existing in the 3:1 ratio. So it is 1/4 of the green seeds are to be combining with 3/4 of the round seeds. This would be resulting in 3, you can say, parts of all the possible combinations. All the possible in the sense, all 16 possible combinations. Okay. Then he went on to explain with the 1/4 of the wrinkled seed random combination. So if 1/4 of the wrinkled seed combined with 3/4 of the yellow seeds. Okay, so these are all possibilities. So if 1/4 of the wrinkled seeds combined with 3/4 of the yellow seeds, again this would be resulting in wrinkled yellow combinations in a ratio 3/16. So of the 16 possible combinations, 3 of the combinations are of wrinkled yellow, because 1/4 of wrinkled seed combined with 3/4 of yellow seeds. Likewise, if 1/4 of wrinkled seeds combined with 1/4 of green seeds. Okay, that is another possibility. So if 1/4 of wrinkled seeds combined with 1/4 of wrinkled seeds, then it would be green seeds, then it would be resulting in 1/16, or you can be saying this would be resulting in 1 possible combinations of wrinkled green F2 plants. So in this way, Mendel was able to prove that because of the independent combinations of these two 3:1 independent ratio that existed in the dihybrid cross, it resulted in a 9:3:3:1 ratio, where 9 of the possible combinations or the phenotypes were round yellow, 3 of the possible phenotypes were round green, 3 of the other possible phenotypes were wrinkled yellow, and 1 of the possible phenotype was wrinkled green. So that is how we have the explanation drawn out by Mendel in order to infer to the results or to the data that he obtained from several thousands of his dihybrid crosses. So based on his dihybrid experimental results and data analysis, Mendel finally formulated yet another significant law of inheritance. And this law is known as law of independent assortment. Now this law states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the segregation of the other pair of characters. Now what this means to say is, when we have an organism, let us say when we have a heterozygote, right? Or when we have an organism having two traits, now segregation of one trait is independent of segregation of the other trait. So let's take an example. When the round and yellow are present together in, let us say, a plant. So segregation of round trait is independent of segregation of yellow trait, and segregation of yellow trait is independent of segregation of round trait. So he means to say here that the two traits are not going to be affecting or influencing each other at the time of segregation during gamete formation. Or this can also be stated as that when we have such kind of two pairs of traits existing in a heterozygote, then each of the allele of one trait can independently combine with the allele of another trait during gamete formation. Okay, so each of the allele of one trait can independently combine, assort means to combine, can independently combine with the allele of another trait during gamete formation. So I'll give you an example. For example, when we have a heterozygote having capital R small r, capital Y small y as its genotype. Now in such a plant, during the time of gamete formation, capital R or the round shape, round seed need not be always going with yellow. Okay, even though the parental genotype is round yellow, but at the time of gamete formation, in the gametes, we need not be always having capital R going with capital Y, because capital R can also it has an equal chance of combining with small y. Okay, so this is how we understand with the independent nature of assortment during the time of gamete formation. So we realized even during our cross that the first R can go with the first Y, or the first R can also go with small y. Okay, and because of which we have two possible combinations of round going with yellow or round combining with green. Okay, in the same way, the second trait, which is the yellow trait, need not be always combining with round. Okay, so capital Y need not be always combining with capital R. It can also be combining with small r. Okay, that is to say, yellow can also be combining with wrinkled. So let's see here. We have this R, that is the small r, wrinkled, combining with capital Y and forming wrinkled yellow combination of gamete. And this small r can also equally be combining with small y, thereby resulting in wrinkled green combination in the gametes. So now, in such kind of the understanding, we get to see that the gametes of the different gametes, some of the gametes are of the parental types. Okay, so you can have round yellow, which is the parental type of gamete, as well as we can have wrinkled green, which is also the other the recessive parental type of gamete. But since we have independent combination of the alleles, it can also be resulting in new combinations, which are actually not present in the parental generations. For example, the combination of capital R with small y, which is round with green, is not a combination existing in the parental generation. Okay, parental generation had either it was round yellow or it was purely wrinkled green. Now here we have a combination of the gamete which can be resulting in round and green, okay, which is a new combination. Likewise, the parental combination was purely wrinkled green, but because we can be also expecting independent assortment of the alleles or one of the trait with that of another trait, so now we can also have a combination of wrinkled yellow. Okay, so some of the progeny can also be expressing wrinkled yellow. We can have wrinkled yellow as a new combination because of independent assortment of alleles. Or we can have a new combination of round and green present in the F2 plants or F2 generation because of independent assortment of alleles or independent assortment of factors at the time of gamete formation. So this law is known as law of independent assortment, which directly explains with how we have the inheritance of two traits in pea plants. So with this, I come to the end of today's lesson. Now I'll see you all with my next video lesson on some of the deviations from Mendelian crosses.